Do changes in cells quicken aging? Can we slow it? I am Edwin Williams, EJW Science Research News. You will like to watch this amazing, amusing, and alarming research. During aging, cellular processes including transcription and RNA splicing, which is a process by which non-coding regions of genes are removed and coding regions are joined together to generate mature messenger RNAs, are impaired. Dr. Cedrics Debra of Germany and his team have succeeded in analyzing genome-wide aging-related changes in transcriptional processes in 5 different organisms: worms, fruit flies, mice, rats, and humans. The average transcription elongation speed, that is RNA polymerase II speed, increases with age in all these 5 species. Along with these changes in elongation speed, they observe changes in splicing, including a reduction of unspliced transcript and the formation of more circular RNAs or circRNAs. 2 lifespan extending interventions like dietary restrictions, lowered insulin-IGF signaling, both reverse most of these aging-related changes. Genetic variants in RNA polymerase II that reduce its speed in worms and flies increase their lifespan. Similarly, reducing the speed of RNA polymerase II by overexpressing histone components to counter age-associated changes in nucleosomes positioning also extend the lifespan in flies and the division potential of human cells. These findings show fundamental molecular mechanisms underlying animal aging and lifespan extending interventions and point to possible preventive measures. Aging impairs a wide range of cellular processes, many of which affect the quality and concentration of proteins. Among these, transcription is particularly important because the transcription is the main regulator of protein level. Transcription elongation is critical for proper mRNA synthesis owing to the co-transcriptional nature of pre-mRNA processing steps such as splicing, editing, and 3' formation. Indeed, dysregulation of transcriptional elongation results in formation of erroneous transcripts and can lead to numerous diseases. But the ultimate question was whether changes in polymerase II speed affect lifespan. Dr. Bayer and his team tracked the survival of fruit flies and worms that carried a mutation that slowed down polymerase II. These animals lived 10 to 20% longer than their non-mutant counterparts. When the researchers used gene editing to reverse these mutations in worms, the animal's lifespan shortened. That really established a causal connection, says Dr. Andreas Bayer, a computational biologist at the University of Cologne, Germany. As animals age, a variety of molecular processes in cells become less reliable, gene mutations become more frequent, and the ends of chromosomes snap off, making them shorter. Many studies have explored effects of aging on gene expressions, but few have investigated how it affects transcription, which is the process by which genetic information is copied from a blueprint of DNA strand to RNA molecules, says Dr. Bayer. Previous research had shown that restricting diet and inhibiting insulin signaling can delay aging and extend lifespan in many animals. So the researchers investigated whether these measures had any effect on the speed of polymerase II. In worms, mice, and fruit flies that carried mutations in insulin signaling genes, polymerase II moved at a slower pace. The enzyme also traveled more slowly in mice on a low-calorie diet. Scientists of this study found consistent increases in the average intronic polymerase II elongation speed with age across 4 animal models, 2 human cell lines, and human blood, and could revert this trend by using lifespan extending treatments. The study also documented aging-related changes in splicing and transcript quality, such as the elevated formation of circRNAs and increased the number of mismatches with the genome sequences, which probably contribute to age-associated phenotype. Furthermore, they observed a consistent increase in the ratios of spliced to unspliced transcripts, that is, splicing efficiency with age across species, which has been reported to be a result of increased elongation speed. Drastic genome-wide changes of RNA biosynthesis would quickly be detrimental for cellular functions and would probably lead to early death. Instead, they monitored a gradual reduction of cellular fitness characteristics for normal aging. Scientists were able to increase the lifespan in 2 species by decelerating polymerase II. Thus, despite being small in magnitude and stochastically, that is, random emerging in tissues or cell population, these effects are clearly relevant for organismal lifespan. Thus, there must be a location-specific factor influencing which genomic regions are more prone to increases in polymerase II speed and which are not. This observation is consistent with earlier findings, indicating that chromatin structure may casually contribute to age-associated polymerase II speed increase. Although they still lack a complete understanding of the molecular event driving polymerase II speed increase, the findings indicate that aging-associated changes in chromatin structures have an important role. The work establishes polymerase II elongation speed as an important contributor to molecular and physiological traits with implications beyond aging. Misregulation of transcriptional elongation reduces cellular and organismal fitness and may therefore contribute to disease phenotypes. Together, this presented data reveal a molecular mechanism contributing to aging and serve as a means of assessing the fidelity of the cellular machinery during aging and diseases. Please also watch my YouTube video COVID-19 and Aging, published 3 months ago, in which Dr. Maria Mavirikaki, a neurobiologist at Boston, US, and Dr. Daniel M. D'Souza of University of Campinas, Brazil, have shown a 10% decrease in cognitive function in the brain of aging in the COVID affected. Thank you. If you are not yet subscribed to my channel, please subscribe.